Hi, this is Alex from devstories.eu, Biopython Fundamentals, episode 16 of 18. You have raw field genotype data and you need statistical metrics, but bridging that gap means writing yet another custom script to parse text files. Converting that field data into a format ready for analysis is tedious unless you use a parser built exactly for the standard Genepop format. That is where population genetics with Bio.PopGen comes in. Genepop is a standard software package and file format used to calculate metrics like Hardy-Weinberg equilibrium, F-statistics, and linkage disequilibrium. Its text-based input format, usually saved as a .gen file, is rigid. It relies on strict line breaks, specific keywords, and glued-together integer codes for alleles. Writing your own logic to extract allele frequencies across multiple loci and populations is prone to off-by-one errors. The Bio.PopGen module avoids this entirely by reading the Genepop format directly into a structured Python object. Take a concrete scenario. You are studying two isolated populations of fish. You have typed each fish at 3 microsatellite loci. Your raw data sits in a standard .gen file. The top of the file has a title followed by the names of your 3 markers on separate lines. Next comes the keyword pop, marking the start of the first fish population. Below that, each line represents one fish, showing an identifier followed by its alleles at all 3 loci. After the last fish in that group, another pop keyword appears, marking the start of the second population. To process this, you import the Genepop module from Bio.PopGen. You open your text file and pass the open file handle to the genepop.read function. This function processes the text and returns a single record object. This is the part that matters. The record object mirrors the biological hierarchy of your study. If you check the loci list attribute on the record, it returns a simple sequence containing the names of your 3 microsatellite markers. This confirms the parser successfully read the header. Then you can look at the populations attribute. This attribute holds a list where each element represents an entire population from your file. Since you have 2 pop keywords in your file, this list contains exactly 2 elements. If you look closely at one of those populations, you find a list of the individuals it contains. Every individual is stored as a pairing of their string identifier and a list of their genotypes. For a diploid organism, a genotype at a single locus is parsed as a pair of alleles. The Genepop text file often stores these alleles as a single combined string of digits like 01, 02. The parser automatically splits this into distinct alleles, representing 01 and 02 as separate entities. You do not have to write string slicing logic to pull the maternal and paternal alleles apart. With the file parsed, you have immediate access to marker names, population counts, and individual allele data. You can iterate through the populations to count specific allele frequencies or pass the record object straight into Biopython routines that interact with the Genepop software. You can trigger calculations for expected heterozygosity or measure genetic differentiation between your 2 isolated groups. The value of the Bio.PopGen parser is not just reading a text file, but transforming a flat list of strings into a strict biological hierarchy of loci, populations, and individuals that statistical algorithms demand. That is all for this one. Thanks for listening, and keep building.